log,
1-2
our
minutes
chat
max.
room, about this association.
That was 14 minutes.
This collection is going to be really informative for us to be able to definitely say what species the Aplocophora
So you saw what I was talking about,
is
right?
feeding on.
With how you grab it.
Yeah.
We don't
Because when
often
you
see
originally
these
had it, you would have had to play all these fun games with the wrist to try to get the tip in.
Scott says that last year on the
Yeah.
Hahana Moana expedition, they saw quite a few.
Same thing when you grab the temp probe or...
Les is saying that he
pick up a
remembers
rock.
when they
You gotta think
started
about-
their coral work a long time
to get
ago.
good
They
at
were
it, you gotta
advised that
think
the
two
most
steps
common thing on
ahead.
the
Yeah, just
colonies
one.
would be the Aplocophorans, because that's what the Aplocophorans eat. And it took them several
sorry,
years to
left
see the
of
first
lasers.
one.
Left edge of screen.
And
Rotating
Scott
left.
Vance says they've never before collected a bamboo with an Aplocophoran. So this is very cool.
We lost about 15 minutes there. We've still got, following the new route now, 560 meters to do.
Video, what am I looking for?
Bottom right.
3 hours.
Oh,
45.
sorry, I see it now. Sorry.
We're still under point 1.
So Shirley, I'm going to go ahead
Just give it
and
a little closer
email
video.
the
We have
two
time for the ship to catch up.
experts for these animals and get their advice on preservation. But I assume it should go in formalin. Would you agree, or you think it's something else?
We were just chatting about that online. We could, you know, why don't you check? I was going to
Yeah, I'm trying to.
toss it in ethanol. I hate to break off a little piece for
keeping that.
OGL, and if we have it in
HD
ethanol, we'll be
view.
able to get some DNA out of it. But so why don't you let me know how you want me to do this?
So that's you tugging me.
Yeah, I
let's
think
be
that's
tugging
important
it.
what you just said, Shirley. If you
Yeah, I can't really
were
come down anymore.
sort
Copy
of
that.
pushed to
All right,
take
video, we're
something
gonna
to put
have to
into a tube for
leave this guy,
OGL,
need to
then maybe
recenter.
taking a wedge from midway along the body so that
starboard.
the head end is preserved, which has some important characteristics I know in the radula, and the back end is preserved, which is where the mantle cavity is that includes the gills or the pseudogills and so on. So other than that, there should be various scales embedded in the body wall, which would be important. But it's probably okay if we cut a wedge out of the middle if necessary, if it doesn't go into ethanol. If it's going to go into formalin, then I think we should at least do that.
Yeah, I guess so. We're going to need to know if we need to have it in formalin to be able to diagnose it.
Pilots, if we can start
And if we
increasing the
do,
angle
then
from,
I'll
say,
definitely cut
275
off a
to
small
28,
piece
285
for
perhaps.
OGL.
We'll
If
shorten
not,
the
I
time
think I'm
down
going to throw the
when we
whole
hit the
thing into
top.
ethanol because it's going to be tiny.
Well, once we hit the top, you can assume we'll do like 0.2.
Let's look at the black
If that
sponge,
helps.
lasers right.
Lasers right, this black bunch.
We're still okay.
Okay.
And maybe just look down on it and slightly fly across it.
Yeah, copy that. I'll do
Is
like
that a different coral on the top right?
I'll make my way there. We'll find out. Alright, video, if you want to come in as a lateral. Let's see what we got. It looks like what it was before, I think.
See if we can
Similar.
actually see some sponge material on the inside.
Yeah.
Well, wait. Is it something else?
Oh, sorry, video.
Sorry.
Little tiny sponges, the white blobs
Yeah,
right around the base
but
of this
this
maybe
bigger
former sponge,
Yeah,
dead
That's pretty
sponge
cool.
Yeah, I
creature.
was trying
very
to.
cool.
Actually, video, can you come out briefly? If you want inside look, I just need to. Yeah, let me get around it real quick and get closer.
You can see if science wants them anymore.
Science, do you have any interest on the inside of this?
No, we can keep going.
Ah, okay.
So spin and look at that coral Josh saw.
Yeah, it should be right to my starboard. Here we go.
Top left of the screen. What is
Is it
that?
left or
This
two here?
Yeah, two different things. The coral's on the right and this other thing on the left.
Washleigh, do you have an interest on right or left?
Let's go right first.
Right first, copy. All right, video.
She likes bubble gum.
Closer.
I do.
I do
I'll
not.
try to get in front of it.
Can't stand chewing gum. Drives me crazy.
I'd like to have a nice thick wad of bubble gum right now.
Would you? And if you blew a bubble, you'd get it all over your
I
microphone.
would get it all over the microphone. just like those larvaceans.
That's the
They have
backside.
a big bubble.
Oh, isn't this pretty?
This is gorgeous.
It's a good side,
This
isn't
is
it?
really beautiful.
Front side is best, but this will do.
Acceptable.
Acceptable.
All right, video, come on now.
'Cause this
Oh,
has-
pretty, pretty.
So anthrax on the back.
Oh, gorgeous.
Oh, wow.
Brittle stars on it, too.
Okay, you might
I
wanna
wonder what
siphon
they've been
this.
eating to give them that color.
You think? You think? Do they eat that?
I don't know.
I don't
I
know.
don't know.
I have no
The
idea.
coral doesn't look as if it's been chewed on by
No.
them.
No.
It's interesting they have a similar color, though.
All right, video. If I want to make it to the other one, I don't have too much
Scott,
time
what did
on
you call
this.
those polyps that are on the stalk that, I mean, the zoids that are on the, are they called zoids? That are on the stalk that don't have tentacles? I forget what they were called.
All right, thank you, video. All right, watch these. I'm going to make my way to the other one before we run out of time at this spot.
Yep.
Just do a snap zoom on it.
Snap zoom. You
Yeah,
got it.
that'll be fine. Yeah, go ahead.
They call them siphonozoids.
Siphonozoids, got it. I'm going to write that
Alright,
down because I
video
know I'm going to
snaps
forget it.
This thing is stalked and very sad.
Okay.
Definitely a sponge.
That's
Exact
good.
and
All right, thank you, video. I'm going to push ahead.
Oh, wow. Look at all the stalks. Dead stalks. This was a forest. Shirley, truly a
This is
forest.
steep.
So the corollary, Chris, was that if we had dived on the southwest side of the fault, would we be traveling through a sponge forest now? If we're seeing all dead sponge stalks here, because the block fell down in this direction on a fault that we can see on the top
Another
of the
single coral
plateau,
straight ahead.
then
Anemone in your lower right.
would there be a forest of these animals
anemone
where
first,
the
sorry,
block
video.
is
I don't think we've
extant
seen as many of those.
and less disturbed? And Shirley puts her
Yes,
hands up in the air.
Yes,
Yeah,
I know. I don't know. I don't
either.
know.
If you sit down
That's
right
the
there,
question.
straight into the mouth.
I mean, how would we know until we get down there and see
Copy that.
what we're seeing
A
and
little
seeing
bit of
all these
time.
dead stalks and sponges here?
Right.
This
But
other guy.
even if we get up to the top of the terrace and get a little further in, we might see some intact colonies that weren't so close to
All right,
the
video,
edge,
if you wanna
perhaps,
come in,
Oh, yeah.
Quick zoom.
that actually survived.
Could be.
Oh, coming up. Coming
Okay,
up.
folks.
Drifting
What
back
is
a little.
this beastie?
Might lose my toe. Alright, video, I need to push ahead.
Some kind of anemone.
Sorry, Washleys, I need to push ahead. You got a quick zoom on it, though?
No problem. Thank you for the zoom.
Video, do you want a quick zoom on this? Yes.
Looks like baby
Drive
bubble
over
gum.
it. Yeah, it looks like the same one.
Maybe.
That.
That's the right color.
Up.
Yep, be coming down on it.
No, it's all good. Little cave.
It's a pillow, dark, shadowed.
It's not a cave.
Nope.
Ah,
Okay. Pilot's ship has been stopped for 3 minutes.
Continue on.
continuing on. Thank you, Nav.
Copy that. Another move, 20 meters, 375.1 knots. Bridge, this is Nav.
Go ahead, Nav.
Same again, please.
20.
20.
20 and
Well, it
275?
is getting
20,
on
275. Good copy.
toward
Thank you.
lunchtime
Thank you.
here on the ship, and I am expecting to see a watch change in about 5 minutes or so, at which time we will introduce you to a brand new
Different
bunch of
thing on the
pilots. But for right now,
yes.
a commercial break, as this is the NOAA ship, Okeanos Explorer, and we are coming to you live from the Mariana Trench National Marine Monument. This is America's ship for ocean exploration, and expeditions like this one, which is the 2016 deep-water exploration of the Marianas, are only possible through successful partnerships and collaborations, and we've got a lot. The mission is designed and supported by NOAA's Office of Ocean Exploration and Research. At the moment, we are looking at a
This one looks a little happier.
rather unhappy sponge. Chris Kelly at the Expedition Control Center in Honolulu. Do you know what kind of sponge that was?
Yeah, it's a tunicate sponge. Sorry, but it's a tunicate. It's not a sponge.
Thank you.
It's a stock tunicate, a really bizarre animal. I think it's pretty clogged up and maybe
having a hard time.
Not certain, but these stock tunicates we identified last year as in the genus Culeolus.
Okay. Well, fortunately for me, Shirley
ahead.
has stepped out to lunch and didn't punch me in the shoulder for calling a tunicate a sponge, which she might have done. Oh, nice little guy.
Down.
Okay, folks at home, in the ECCs and experts on critters, tell us what we just took a zoom on.
Let's see. I think I saw a bigger one.
I think we were looking at a Chrysogorgia octocoral with a squat lobster, probably family Chirostylidae, in its branches, and then 4 little sponges on the rock to its right.
Thank you.
Is this what we're going
Scott.
towards?
No.
No.
We just looked at.
Oh, so we just looked at. I can back
On the
up briefly.
right of that one is more interested.
So to the right.
Yeah, just
Right
past
now?
Yeah, just past the candelabra one.
Ah, okay. Oh, back there. I see it. Yeah, video. I will get closer if you want to come out briefly.
That's the one at the bottom of the screen.
Oh, they do want the one at the bottom.
I don't know if they want it.
Oh,
It's
I'm
just-
sorry, watch leads. Which one are we going for?
It was just something I wanted to show them to see if they wanted to label it.
I guess we want to have just a quick snap zoom on the one in the upper center.
Upper center, that is. Okay, copy that.
There's something very red on the bottom right. Maybe an octopus. Just went past your skid.
Ooh,
Okay.
we should take a look at that too. Yep.
Copy that.
Pilot,
I will
take a
look
look.
for octopus.
Our videographer thinks he may
Maybe a-
have seen an octopus.
I saw the shrimp. I didn't see an octopus.
Right hand edge. It's actually one of those coral balls.
the red
That's-
on the
that's
left.
Not an
Yeah.
octopus.
Not an octopus.
But
Oh,
an
shucks.
octocoral.
We shall take a close look at this little red guy sticking to the side of a
Yeah,
rock.
video, that's kind of far away. I have to get down there
This
for a
looks
better
like,
view.
this is an Anthomastus octocoral,
right
and
there.
this is nice because we've seen
Translucent.
several
Oh,
of
yeah.
these now
What
with
is
these
that,
very large
wow.
polyps sticking
I
out.
don't remember what they're called.
And I earlier on another dive described it as the base of the colony kind of
video,
being like
come on
a
out.
squeezed fist
Nav,
with the wrist coming
co-pilots,
out
or-
the bottom.
Hey,
So
co-pilot.
there you can
Hey.
see all those
How you
dark
doing?
Hey, just
circles.
checking,
That's where the
are we
polyps
in the
have
middle
retracted
of a move, or
inside
are
the
we
matrix
stopped?
of the colony
We are in the middle
itself.
of a move.
And then
Yes,
now we're
we're
looking at one of those
about
tunicates
5
that Chris
meters to
was
go.
just referring to, but this
5
one's
meters to
alive.
go on the ship.
These are
and
unusual
heading
tunicates
was
in that they're
generally
carnivorous. They have a kind of a large
275-ish.
hood,
It's
and they're
close.
able
We
to
can
trap
adjust it
zooplankton in that
280,
hood and
right?
feed on them,
280,
whereas
okay.
most tunicates are more
right
like
now?
suspension
We
feeders.
just, oh no? Oh, my fault. 275. My mistake.
Thanks, Scott.
Roger that.
Alright, video. If you want to come in.
Oh, so this is the tunicate.
Doesn't hide anything, does he?
No, not at all.
Very translucent. Carnivorous beastie.
Stabilize in center anymore.
Yeah, copy that video.
And that loop that you're seeing down below, it's brownish and white, that is likely the digestive tract. Not 100% certain. And probably that whitish, berry-looking stuff is probably gonadal tissue.
Oh, thank you, Scott. Quite a lovely image.
Yeah, we're looking through a hood, and there's a stalk in behind it that goes down and attaches itself to the rock.
A little bit.
There are lots of tunicates in shallow water. People are probably familiar with them called sea squirts. And so this is just a very aberrant sea squirt of the deep that no longer suspension feeds, but instead in these low food environments has adapted this ability to trap swimming zooplankton.
Ah.
Pulling out.
Cool.
Lasers.
Copy that. Co-pilot lasers, please.
They're on.
Lasers?
Oh, they're on?
Yep.
Never mind. I'll rotate up.
Okay.
Okay.
So it's about 10 centimeters wide. Ear to ear.
Full wide.
Copy that video. Let's see, do we want a view on the red item to the left?
Well, if you're stable and have the time.
I think we have a little bit of time, right, co-pilot?
Yeah, I believe so, yep.
Copy
You
that.
can do that.
Oops, sorry, video. I just needed to did a little bounce step.
Just trying to get another quick look at the structures on this that Scott was just describing. I think we did see one of these earlier with the
Tilting down.
tentacles extended.
Yeah, tilting up.
It's a nice view.
That's right, Patty. In fact, the entire polyp is extended.
I
Were
see.
extended, the ones that we saw earlier. So here, all the polyps are retracted. You can sort of see those
You very
star-like
slowly rotate
formations,
left, see the other
those
structures.
swellings. That's where the polyp
Rotating
would retract.
left.
And when it's
Slowly.
out, you would see a fairly long
That's good. Hold
polyp
there.
extending upwards, a little tube, and then the tentacles would come off the end of the tube. And then all the little white bumps are miniature typhonozoid polyps that don't have tentacles.
Okay,
Oh, that's
pulling
cool.
out. You can lift off.
What a complex
Copy
beastie.
that. All right, and
Very
watch,
likely that
do we
little
want a
sort
quick
of
look
elongate,
at
sort of volcano-shaped thing to its left
this right
is
here.
another one that's
Coming
growing.
center.
And you can see coming from the right that looks like a tail. That's probably tissue elongating from that colony that could possibly put up a new colony later on, asexually.
Thank you, Scott. Yep, I think we're going to have to get moving here, but we just zoomed on another
Yep.
bubblegum coral.
You want to start pushing out, pilot, please?
Yeah. All right, wash leads, I'm going to push ahead.
Okay.
Yes. Thank you, pilot. That's great. Just to let you folks know who puts these expeditions together, the mission was designed by NOAA's Office of Exploration and Research, OER. The ship itself is operated by NOAA's Office of Marine and Aviation Operations, and the expedition that we're on right now is part of a 3-year campaign to address the Pacific monument science, technology, and ocean needs, and its nickname is Capstone. So you'll hear us talk about Capstone on occasion. It was designed to address the fundamental need of the resource managers to better understand the deepest parts of the nation's marine monuments. Some of the key partners in this effort are the NOAA National Marine Fisheries, NOAA Marine National Monuments Program,
Lift off while
and
you guys are
NOAA's Deep Sea
changing out.
Coral Research and Technology Program. Another important partnership for the Okeanos is with NOAA's National Center for Environmental Information. And through this partnership, all the data that we're collecting are publicly available within about 60 to 90 days after they're collected. So this is very unusually fast for data to be made available to the public. Normally with a federally funded research expedition like the one we're on here, it would be 2 to 3 years for a moratorium period so that the scientists who participate can publish papers, finish their analyses and so forth.
Something white.
So everything that's happening on board this ship is made possible through a very close partnership with National Center for Environmental Information. And the samples that we are collecting out here will be archived, both the rock samples at the Oregon State University and the biological samples will be archived at the Smithsonian.
Pilot, good afternoon. Looks like Sean had just started to move. We need to push out a little bit and probably get a move underway going up.
Has the ship stopped now?
Of people who are operating the ROV and navigating us.
Couple minutes, yeah.
And they are
we were just catching up, and I think
engineers
we
from the
finished
Global
here.
Foundation of Ocean Exploration. We'll
They
give
were
them a
moving
chance to
275
introduce
at about
themselves
the
once they get
10th.
themselves all settled here.
Yep.
It looks like in high pack we're almost to the flat on top.
That's what I was told too. Yeah.
All right.
And then I believe there's a waypoint going up to the north somewhere that we need to shoot for.
Yeah, I heard that. Can you zoom out now?
Yeah. So.
Hey, watch lead. Did I hear something from you?
Yes, pilot. We would love to hear you introduce yourselves up there in the front row.
Okay, going to OKEX. So we just had a shift change. New front row here. We're a team from the Global Foundation for Ocean Engineering. Engineers, we designed and built these ROVs. Now we maintain and operate them at sea. I'm Carl McCletchy, the co-pilot. On my left.
Levi Unima as your navigator today.
And on the right.
Don Liberatore as co-pilot.
And then really far right.
This is Roland Bryan. Video.
And in the back we have...
I'm Annie White doing the video clipping today.
So we'll be driving around for the next 2 hours.
Thank you, folks. These guys are great. As you have seen, all of them are great. And we'll be having a wonderful time.
Yes.
Right now we are diving on a flat top undersea
So the name of the game
volcano that has
is up the ridge,
had been erupted
and then kind of over to dive.
probably somewhere
so
between
keep going
100 and maybe 20 to
If there's any rocks, we'll follow
gee,
it. We
maybe
do not want sediment, I think.
80 or so million years ago
That's the name of the game.
on the Pacific Plate. And the dive today was
275
chosen
look
by Chris
Lots of
Kelly
stuff
at the
on
University
that,
of Hawaii. He's in
dead
the
branch.
oceanography department there, but he is also in charge of the capstone 3-year expeditions.
Video swapping
And maybe, Chris, you could give us a quick introduction of yourself and why you chose this spot.
Sure,
Did
I'm
we get
Chris
a move
Kelly. I'm
underway?
with the University of Hawaii here in Honolulu, Hawaii. I'm serving as a science advisor for OER, and I'll also be the onboard biologist for the next cruise at Wake. I'm the one who submitted the proposal for this site. It's on a manganese-crusted geode, as you can see on the video right now. These rocks are very heavily manganese-crusted. This is important because this type of crusting will be the target of deep-sea mining operations in the future because they're very rich in lots of metals and other minerals that we need for our technology. So we like to survey these type of substrates and these type of seamounts that'll be targeted and try to understand what the communities that are living on these crusts are like and who's vulnerable, and you get an idea of areas where there's not very many animals versus areas where there's a lot of animals, which may require some type of protective measure.
Thank you, Chris. And we are now looking at a fish. And today you're also the fish man, right?
Yeah, I'm the substitute for our normal superb fishman, Bruce Mundy, and some of his knowledge has rubbed off on me over the years since he's here in Honolulu as well.
So what is this guy?
I'll take a bit more zoom.
All right. Try to get
So
ahead of
this guy is another cutthroat eel in the family Synaphobranchidae, but he's different than the ones we saw earlier.
Yep.
And I got to look at him for a second.
The eyes are a lot bigger.
Okay.
I do not even see his dorsal fin, so I guess it's way back. It is way back. So this may be the true Synaptobranchus brevidorsalis. I'm looking for a fin on the top of its body and where it originates, and you can barely see a little line about 2/3 of the way back on the body, and that's what I was looking at.
Wow, yeah, you can hardly see that fin on the top of his body. Just there it is, just coming into view. Very, very thin.
Okay, we'll let him go. Thank you, video. Watch lead pilot.
Oh,
yeah. What else are we looking for out here? Anything in particular?
You mean just in this area or in general?
In general for the dive, are we hoping?
I wonder if we could look at this coral straight ahead. This is the bamboo
this
that I
coral
don't
straight
think
ahead.
we've imaged today.
That's a good start.
It's good. Gives us a chance to catch up here.
Okay, I think we're pretty much very close to the top, so no problem there. This is bigger than it looked from the front. Okay, video, free to zoom.
So two of our shore-based scientists, Scott France from the University of Louisiana and Les Watling from the University of Hawaii, work on these bamboo corals, and I believe they're doing a revision of them, and they're finding a lot of new genera and species.
Co-pilot, will you turn the lasers off?
Sure.
That's right, Shirley, and this is one of the new genera that we are describing. Has this very characteristic kind of candelabra growth form, and you'll see, if you look at that sort of lowermost branch, as new branches arise, they are arising where there are these dark spots. Those are called nodes. They're basically protein, and in between them is what's called the internode that's made of calcium carbonate.
Okay, can you tilt to the top where the branches end? Perfect.
That's good.
Just out of interest, I'll note that it was
I think I stopped again.
bamboo coral like this candelabra, this new genus, that was the first to have its mitochondrial genome completely sequenced.
Oh.
That was by a former PhD student of mine, Marcia Brugler.
Hope you're not getting seasick over there, video.
No. It'll take more than that.
So any of these ones that are the candelabra-shaped ones are ones that are your a single new genus or multiple genera? And I guess I have two questions there. One is the candelabra, that morphology, new, and is it a single genus or more than one?
Right, so there is currently no described bamboo coral that has this candelabra shape. And as far as we know, all of them belong to this same genus. At least the ones that we have collected and done the genetics for, they appear to all be the same species or maybe two closely related species.
Yes, please.
So right now we're focusing on one genus that has this sort of shape. And when it first starts growing, it doesn't have any branches at all, so it just looks like a whip. And then when it first gets its branches, it looks like a little trident. And then later on, when it's much older, it takes on this appearance that we're seeing here. So depending on when you see it, it could actually look quite different.
Oh, that's interesting.
I mean, when you see it relative to foul.
Yeah. So is it possible that some of the little whips that we've been seeing are just younger forms of this genus?
Yes, surely that is entirely possible, and that's one of the reasons why, when it's possible, we like to get a close-up look at the polyp, because then we can try to use the position, shape, and number of sclerites as some informative characteristic to compare it to what we were just looking at here.
Yeah.
Of course, ultimately, as you've said many times before, it really means having it under a microscope.
Yep.
We've gone by it now, but Les was noting how interesting the way it grew out of the rock. The larva must have settled on the lower part of the incline of a rock, grown outwards a bit, and then turned and grown upwards. So it took on a really interesting shape.
Yeah, kind of like a creeping branch at first and then went upright. Go ahead and get a snap zoom on this, please.
Okay, want to go in video?
Okay, so this is a smaller of the bubblegum corals that we've seen.
Okay.
The brittle stars attached to the branches. And it also has zoanthids, so zoanthid polyps that are on it. So some of those lighter colored tentacles that you're seeing are from another cnidarian
I
that's
should probably get a toe
growing
down.
on the bubblegum
Just hold
coral.
there. I'm almost there. It's never really a snap zoom, is it?
It's a bit of a paradox. Every now and then it is.
That's
rarely,
good.
Rarely. Okay, got a toe down. You can go in video.
Oh, thank you.
All right, so you can see the different polyps there. So you can see the tentacles from the bubblegum coral, but then over to the left of the branches, you can see the zoanthids that are attached. Have much longer tentacles.
Okay. I'm going to rack focus back to the stars.
It's strange, Copilot and Nav. I keep disappearing off the right side of the screen. Almost like the Sirius compass is off.
Didn't they report something like that on launch?
No.
At first, yeah.
Oh, they did.
I think so, yeah.
That the compass was off? All right.
Possibly.
Whenever you're clear video.
We thought we straightened out during ascent. Or descent, excuse me.
It just seemed really far off.
Yeah, like it was 15 or 20 degrees.
I thought I heard the pilot this morning say 30.
Sure. Is it 30 all the way?
30, yeah.
Big one.
But that was right on launch.
This is the same as the other one. We could skip this, Shirley, and keep moving up.
Okay.
So really,
yes,
you need to come around more to
we can just skip
starboard.
this and keep moving
it
up.
seems that would help. Because
come around, yeah.
You should have been heading straight at me, which
some more of these.
I haven't really moved at all. So
tubular
so
structures
what's the direction
that
of the ship?
may be
heading 275, right?
Yeah, 275 is what we're moving.
With
Okay,
manganese.
so you're
That's
20 degrees to starboard
It's a
now.
wild guess.
That's a lot. It's kind of odd. Kind of makes sense looking at head pack. Really, upslope should be
more like 300 or so.
Yeah.
Well, let's see how that works.
So
I can get here and align with you and see what it looks like. Oh, but I should be...
Yeah, that's right. Let's compare that. See if you can get straight on.
So we should be getting close to the top of the slope, and this looks like it might be, could be there.
Yep, we are at 1,965.
How far along are we in the move? Did they stop, Nav?
Yeah, ship stopped 5 minutes ago.
No, we should start him up again, eh?
Yeah.
Yeah, where's that next waypoint? That is...
They want us to keep going.
Keep going this direction, uphill.
And then kind of take a right towards the dive end here.
Okay.
So go up.
I'm actually headed 310 also, and it seems to line up.
Yeah.
It's odd that...
So there's a yellow fan that's behind that sponge. I wonder if we could take a look at that if he's going that way.
It's just the current pushing you off that way?
Okay, let me just check and see. I'm not sure I see what you're saying there.
Look at Sirius's tracks.
Carl, one of our shore-based scientists says he sees a yellow fan just behind the sponge. I'm not sure I see that, but...
It's behind the boulder that's behind the sponge. It's right in dead center right now.
But behind the boulder?
It's about to go out of view because he's drifting down and to the right. It's just poking out from the left side of the boulder.
Directly above the white sponge in the middle.
Okay.
So what direction of movement are you guys thinking for next?
Well, I lined up my compass, and it was also 310.
Oh, yeah, okay, so I'm seeing it now, Chris.
Somewhere around there, 310?
Sounds good.
Okay. 30 meters, sound good?
Okay. Maybe even 40.
Of course, it may not be a yellow coral. It may be a dead sponge.
Yeah, I think it's a dead sponge.
I don't know.
I'll make my way over there, just waiting on Sirius.
Corkscrew stems
30 and then
that's
we'll
covered with manganese.
Okay,
I
we can add on.
know what that is.
Yeah,
Do
they
you think
might
that's
wanna...
a Faria sponge?
Bridge ROV now.
Chris, that's just been a dead Faria that's been encrusted?
Ready for a ship move, 30 meters.
Yeah, it looks like it to me, Shirley.
Decimal 2 knots.
I'll come down a little bit and give you some more leash.
Little sponge, some creature on it.
Sure.
Of course, it's directly behind that one. Let me hop over.
So I copy 30 meters, 310.2.
Good copy.
Yeah, what we're interested in looking at is that yellow fan that's behind. So
yeah,
we can,
just waiting on Sirius. Okay, video, we've got 2 in frame. You can zoom and focus as you'd like.
Sounds good.
This,
Right.
So that move is a minute or 2 in?
Yeah, just call it in.
Alright, thank you, video.
Of course.
Make my way over to the
There's
yellow
a whole
fan.
little community on that little dead sponge.
so this looks like some of these dead things that we've been seeing on their sides.
With a bonus coral at the base.
With a bonus coral at the base, indeed. Wow. What do you think, Chris? Giant paddle sponge?
Okay, want to get lasers off, Copilot?
Roger no, I don't think so. I think this is actually a dead Lithroyella. And I'm going to wait for him to zoom on this, but the live ones typically have a commensal anemone associated with them that's all over their surface. So I I'm not seeing it on this guy, so I'm sort of imagining that when the sponge dies, so does the anemone.
Yep. How about if we zoom on the base and then kind of tilt up towards the top? We can get both of these organisms as we do. All right. I'm going to just hold there.
So you see these little round tubes that look like oscula on the surface? You have some smaller irregular ones, but then you see these very round tubes.
Yeah, is that where the...
these are where the anemones lived in the sponge, and presumably they caused the sponge to secrete these tubes around them. And so every single one of those larger round tubes is where an anemone used to stick out.
Perfect.
There's the top.
Lovely.
Anything else of interest here, Watchley?
No, this is good. Thank you, Carl. So, so Scott was just wondering if this was, when we saw a little bit deeper, when we saw the kind of a fan-shaped dead
Thank you.
something lying on its side, and we were wondering, or I was wondering whether it was a dead sponge stalk or a dead coral.
So this time I have state center screen. Maybe a little current
this gives us a
because
little
I
bit more
was
of an
feeling a pretty
indication of what
strong
it
current
could be,
as I
except
came
I'm
up
not
over
sure that the way the fibers are arranged, the spicular fiber tracks are arranged in the sponge are, you know, kind of like dendritic like that, that would leave that kind of a skeleton.
So do you see how many dead stumps are here? Tons of dead stumps all over the seafloor here. So I don't know if we're just witnessing a community that was changing over a very, very long time, or whether there was some event here that caused a catastrophic fatality event.
How far has that move gone, Nav?
27, 28 meters.
28 out of 30?
Yeah.
You want to tack on another 20 to it, or another 30? Or?
Well, you probably haven't started moving yet.
It looks like we're seeing a
called it in at 22.
Another
22.
bamboo
Or you
coral
started,
and a
but
chrysogorgia.
I'm pretty sure we're
next to
moving
it.
closer to you. But
An accumulation of these dead, probably sponge stalks with manganese coating.
Yeah, pilot, you okay with tagging another 30 on, or you want to hold off?
I'd wait a couple minutes till it really settles out.
Okay.
I just don't like stacking up to 60 because then when they want to sample,
yeah,
then you're in trouble. You're right. Yeah.
And I don't think we're in any. I think we're already at the top, so everything else we do is
okay.
Yeah, we're not in a rush to get
lasers back on.
Thank you. This thing over here looks different. Will you zoom center screen? I guess it's not that different.
Bonus anemone.
So ship's in position, so about 5-6 minutes maybe, settle out.
Yeah, you can see how much closer he got just as we talked about it.
Yeah.
Excellent. As long as I'm here, let's go in on this anemone.
All right.
Sorry, guys, I can't see the event log. Don't ask me why. I can't. So let me see. Join group chat.
I don't hear any talk either.
Yeah, I know. There we go.
All right. There's lots of different
Diva's
things
calling this an Anthomestus. Actually, Scott, Diva, and Tina are all calling this Anthomestus.
Before I take off from here, is anyone interested? I see four other...
So what I'm thinking is that we can just keep pushing in over the top of this geode and see how far in the hard layer extends until perhaps we get into some sediment and see what the actual distance of that is.
Okay. Let me communicate that to pilot and nav.
Nav, did you copy that?
Yeah. Push in. Keep going on and up, I think.
Yep, on and up over the flat spot, looking for the interface from hard bottom to sediment, sort of.
Copy that. Pilot, you okay with another 30?
Yeah.
Okay.
All right.
Oh, this is pretty. This is Narella. This is another Primnoid, possibly Narella.
It has so much
well, let's see,
detail.
we
It almost
have
looks pixelated.
Bridge,
Chris
RV
is saying Narella. Asako is saying
can we tack on
possibly
another 30 meters
Narella.
to that last
Very
move?
pretty.
30 meters, 310, 0.2.
Good copy.
I don't know if the push is going to be that far.
Yeah, really.
All right.
There's sediment.
I pushed to the sediment.
A push to the sediment.
That wasn't very far in before we get
Yeah, about that, right.
What's the
Seek
next move?
and you shall find. Here we are. We are at 1,962 meters.
There's still some scattered.
Could we take a look at what's all over the sediment bottom? It looks like coral skeletons or something. I don't know what that is all over the bottom.
We want to take a look at what's on the sediment.
All right. Yeah, there's
Please.
a lot of it. I'll come down a touch. Okay, video. Free to zoom.
Okay.
Looks like more of those stalks that we've been seeing, except they're either buried or broken.
Looks like Mount St. Helens after the explosion.
Yeah.
Okay. That's a good overview.
Okay, so what are these ones that are more like mound-like? Is that, see how?
That's rock.
Is it rock? Is that just rock there? Not just rock.
Video?
Rock.
Yeah.
Sorry.
It's amazing.
Yeah, just looks like rock. So we're not really in it. We're still in the rocky part of the geotop.
Okay.
Yep, that's all rock. So we haven't seen any of these large stalked creatures alive?
No.
Something upper left looks like something sticking straight up.
Yeah, I'll go there first. Quick zoom in the middle video. Okay, that's good. It's just a shadow under it. Different.
Happy to check. It's cold. Shirley and I are freezing in here today. I can't find the arm of my jacket. There we go.
There you go. Oh, pretty.
Thank you, Jim. I'm going to punch you now. Ah, there we go. Fussy
Warmer
jacket.
now?
Oh, yeah, maybe. In a few minutes. Oh, boy. Look at this beautiful coral up there.
I know, isn't that pretty?
And
aren't they
bubble
pretty?
gums.
Yep. Bubble gums.
Now, is that bubblegum coral. Oh, okay, there are two of them there. Wow, both of them bubblegums. Wow. Very nice.
Oh, and the one over on the left is Trissopathes. So far left is a black coral, Trissopathes. The two in the center are the bubblegum coral, the Paragorgiidae. And the one just above it, what are we thinking?
Looks like an Iridogorgia to me from here, but I'm not
Want to zoom in video?
Sure. Oh, wow.
Take a bit more. Right there is.
It does look like there are 3 polyps that come out of the same level, or are they spiraling? No, looks like they're coming out of the same level.
Yeah, looks like you're right, Patty.
I know.
Okay. Looks like
Somebody mentioned. I think Tina mentioned that way earlier in the dive. Almost the first coral we saw looked like it had 3 polyps coming out. Is that different from
and
most of them?
Scott actually said polyps, 3 per whorl.
Okay. So that's not unusual.
They're returning.
Okay, yeah, now we're getting into rockier areas.
Oops. Well, I just wrote in the chat log that it was more sediment covered and less bumpy, and now it's back into bumpy.
As you take your eyes off the
I know.
monitors
You start writing
for like
stuff down and
10
it just
seconds
changes.
to type something into the event log.
How dare it?
So this is the same Narella. We could probably skip a close-up on this one and keep going.
Okay.
Okay, did you hear that, pilot?
I did.
Ship just got into position.
Excellent. So now that we're on this flat top, there's a bit of a current from starboard to port or north to south. Not too much though. I want to zoom my sonar out to 30.
So we have about a little under 3 hours left in this dive.
And we are at 1960 meters or so.
So that's straight ahead.
Looks like a big stock. I was going to look at this bright white thing and go over there.
Yeah, okay.
Pilot sees a bright white thing.
He's moving
Starfish,
over to it.
Yep.
Perhaps.
Not sure.
On the seafloor on the side of a little
Yeah, maybe a
mound
little
that
starfish.
probably was a rock before it got totally covered with manganese coating.
Now I know that we never snap zoom on a starfish, so I'm just going to set down.
SOP.
Looks like a nice anatomy over there, too.
How do you know it's nice?
Because
Its little
good
arms
size.
are up into the water.
A stalked fuzzy thing behind it.
Oh.
Okay, video.
And the red dots are 10 centimeters apart. And this looks like a sea star.
It is. It is indeed.
Let's see who it is.
With its little arms up in the water column. You could see the tube feet.
Zoom just a touch. There, that's
There we go. Now you can really see those 2 feet.
I do not see them moving. Maybe it's just sitting there.
Yeah, not a lot of movement.
It's a very stationary starfish.
Sitting,
That's
digesting.
Good. Can we look a little bit to the right, please?
There's an anemone in the
There's
back.
a
I'm
big
not sure
anemone.
I know, but we want to look at a coral that's over to the right, too, that I think we may have just passed.
Okay.
Not the one right in
No, not
front.
no. There's a chrysogorgid
Okay.
that was a very large one. Is it the one in the distance that you're thinking?
That looks like a sponge to me.
That looks like a sponge to me,
I see
too.
yeah, I think I see it.
It looks like that one looks
It's
like
very
more
hard
of a
to
black
see.
coral, though. From here, I don't know.
Wow.
Oh, I see.
Okay, I'm gonna go in.
No, I don't think that's... Is that it? Oh, yeah, maybe. Wow.
Good eyes.
Very delicate. All kinds of other things crawling on it.
Okay, I'll zoom just a touch more.
Yeah.
Is this chrysogorgid?
I'm not sure if this is the one they were talking about, because they said there was a large one. This is this is a chrysogorgid, but I don't know if this is the one that they were talking about. Very delicate branches.
Whenever you're clear, video.
Alright.
We're going to go for the stalk sponge now. Or
Yeah, I'll back off a little and see if I see a large coral.
okay.
Was it to the right, Shirley?
Yeah, that. I believe Tina said there was a large one over to the right, but perhaps it was the thing that we're thinking in the distance is a sponge. Not sure. Okay.
Well, yeah, I don't see any
Yeah,
other corals around.
I don't
so.
see it either, Carl. Let's move up to that the large
The
stalk.
sponge?
Yeah,
The stalk?
Yeah, I'm not sure. I don't know
Stalk.
that that's a sponge. It might be that might
Okay.
be what Tina was referring to, actually.
Okay.
Hello.
Hello.
I noticed you saw a starfish. Hello, Chris Ma here.
Hi, Chris. Can you just stand by for?
Starfish.
Yeah, just stand by for 1 sec. We're just moving in on something else, and then you can talk to us. Yeah, we missed you.
We would love to hear what you think about the star. As soon
So
as
we're
we're
going to look
done
at
with
this.
this,
Yeah, no, I think this is a
looks like it has spicules all over it.
Well, this is really interesting.
Here in front of Chris and point out this is a Walteria sponge. It's a glass sponge, and it's Walteria CF lucardi. Very beautiful and a live one.
So, Chris, do you think these are the manganese-coated dead stalks that we're seeing?
Okay.
Because the size looks about the same.
Can we turn the
It just seems like it's a very delicate stalk, though.
Yeah. You can turn them off now, Copilot.
Yeah, I don't think it's the same thing.
Yeah,
And actually, will you take my camera and
this.
tilt up?
Yeah,
Sure.
really.
Thank you.
Okay.
Just follow video's cue.
I just told you.
I didn't copy tab.
There's
Tilt
a
up
brittle star
Tilt up slowly.
to the left. And
Continuously.
what is
How's
that?
that?
A shrimp?
Yeah, it's a
Perfect.
shrimp in
Yeah,
the
just in the
background. Yep. Yep.
And then just
See,
keep
we got to
going till
quit
you get
abusing
that.
Chris Ma and let him describe the sea star.
Yeah. Okay, Chris, why don't you tell us about that white sea star that we saw just a couple minutes ago?
Sure. So that is in the genus Anthenoides, Anthenoides. And it's a goniasterid. Goniasterids are some of the most abundant, or I should say the most diverse sea stars at depth. They're on the continental shelf and so forth.
I think we've often seen a lot of these, some of these white nondescript kind of heavily sort of armored animals.
Fairly large.
The reason this one sort of rings a bell with me, though, is because this animal,
He's got a first-class seat up there, man.